Thank you. Good afternoon, everyone. Thank you for sticking around. My name is Stavros, PhD student from Delft University of Technology in the Netherlands, in a joint project with a Dutch company called Kijin. And what we are trying to do in this project is come up with a computational method that will automatically assign gene ontology annotations for plant proteins, specifically for plants, and we are mostly interested in the biological process ontology, which, as we saw already, is, happens to be the most difficult one. So we want to have a method that will get a plant protein that has no annotations, and then accurately be able to assign all the Go terms that correspond to that protein. There are, of course, a lot of methods that try to tackle the same problem, and here you can see a picture from the GAFA2 that shows the performance in Arabidopsis thaliana in the biological process ontology of the 10 best methods. So you can see 10 gray bars, and the height of the bar is the F measure. And also the blue bar on the right and the blue line shows the performance of BLAST in the same dataset. And the difference is not that big. So not only between the methods, but also between its methods and BLAST. So our idea is that function prediction, and specifically in the biological process, needs a leap. We need to find a way to take a big step forward and improve performance. And we actually had two ideas for that leap, and I will discuss today. One concerns a new feature representation of proteins, and the other concerns a transformation of the Go terms. So let me start with the similarity representation of proteins. If I have two proteins with high sequence identity, then it's usually easy to infer that they also share the same function, and BLAST is very good at identifying these cases. But it gets more difficult as the identity goes down, what has also been called the gray area of identity, where sometimes proteins with low sequence identity share functions, sometimes they do not. And we want to improve performance specifically in that gray area, so we don't use the similarities to only the top hits, let's say, but we use the similarity to every available training protein. So to give you an example, imagine that we have a database with 3 annotated proteins, we call P1, P2, P3. Then we define a 3-dimensional space where the x-axis has the identity to P1, the y-axis identity to P2, and so on. And in this, every point in this space is a sequence, and the other way around, every protein sequence that you can imagine is a point in this, inside this cube of length 1. Now, if you do the traditional approach and only look at the significant hits, so let's say I had a protein PA that is 95% similar to P3, and I blast this protein, then I know that it has 95% identity to the top hit, so it must lie somewhere on this red plane that I have drawn here in this cube. I could have a second protein that also happens to have 95% identity to P3. So this second protein lies exactly on the same plane. But if I only look at P3, if I only look at the significant hit, then I am forced to predict the same annotations for A and B. But actually, if you look at the gray area, you see that A is much more similar to P2, and B is much more similar to P1. So we say that maybe this slightly different similarity profile might imply that there's difference in the functional profile of these proteins as well. So given a database of annotated proteins, we take each protein and calculate its sequence identity to every protein in the database. And this gives us a similarity profile. And then we use these profiles as feature vectors, which then feed into a classifier. And the classifier based on the distribution of these feature vectors, can learn which proteins have a specific function and which do not. To test this feature representation, we use the k-nearest neighbor classifier, and we named this approach SimKNN. So that was idea number 1. Now idea number 2, which is based on the redundancy of the Go terms, which has been mentioned several times in this meeting. So if a protein is annotated with a specific term, then by definition of the hierarchy, we know that it is annotated to its ancestors as well. And if you combine this, that with the huge number of terms that are available in the ontology, especially in the biological process, then we think that maybe the classifier's job is too difficult. So we try to make it easier for the classifiers. We try to exploit this redundancy in the gene ontology, reduce the number of labels that the classifier has to learn with the hope of increasing performance. To put it a bit more formally, if we have a feature matrix X, so in the rows the proteins, in the columns the features, and a binary matrix Y with the labels, so the rows are again the proteins, and the columns are the Go terms, then instead of training a classifier from X to Y directly, we first take the matrix Y and transform it into a lower dimensional space, which we call Y prime. Then in this transformation, the Y prime is not necessarily binary anymore. So we train a regression function from X to Y prime, so our new target variables. Now, if I have a new test protein that I want to annotate, which I represent by its feature vector little x, I can put it into my regression function, and the regression function will predict the values in this lower dimensional space. And if the mapping that I chose at the first step is linear, then it's actually easy to go from this lower dimensional space back to the Go term space and get a new feature vector with as many elements as Go terms. And every element here contains something like the posterior probability that my protein X is annotated with that Go term. This is not a new concept. It has been used in other domains. And one of the most simple methods to do this that we tried is called PLST. And it is essentially nothing more than principal component analysis on the label matrix Y. So you map your Go terms towards directions in the label space that have high variance. There was an improvement on that method called CPLST, again based on principal component analysis, but on this more complicated matrix that you can see here. Notice that it contains X as well as the Y, and it's easy to show that actually what this method does is you say, okay, I still want to have high variance in the labels, but I want my target variables in the new space to be easier to predict. So I want, when I do this mapping, I want a more easy classification problem. So using this expression for X inside, it pushes the Go terms towards directions that are easier to predict. We were a bit inspired by this method, and so we made a different method that actually takes into account the fact that the labels are organized in a direct acyclic graph. So we make a binary matrix G, which is square, and has as many rows and columns as there are Go terms. And now an element ij in this matrix is 1 if term I and j are parent and are connected by an edge. So and this matrix is symmetric. Now if we apply PCA in this matrix Y transpose G transpose GY that you can see on the screen, what we achieve is a mapping that again looks for directions of high variance, but also tries to push related Go terms towards the same direction. So it is trying to maintain the hierarchical structure. So to test these ideas, we took 8,000 proteins of Arabidopsis thaliana from SwissProt, all with annotations in the biological process, excluding electronic annotations, and tested in a threefold cross-validation loop. We tested our similarity representation with k-nearest neighbor against the multi-source KNN algorithm. And so we test these two methods on their own, and each of them combined with the three-dimensionality reduction methods that I talked to you about. And we used two baselines, simple annotation transferred by one blast hit, and also the variation of blast used in the CAFA experiments, where annotations are transferred from all the hits at different levels of confidence. For completeness, let me quickly go through to our competitor method, the MSKNN. So if I want to predict if protein P should be annotated with a function f. So I find the K proteins in my database that have this function f, and if one of these proteins has this specific function, so it is the identity function I in the formula, then this protein votes in favor of P getting that term. And the votes are weighted by the similarity between P and its neighbor. So we compare against this method, which was one of the best in CAFA 2 and I think also in CAFA 1. These are our results for the maximum protein-centric F measure. So the blue bars show our similarity representation, the yellow bars MSKNN, and so you can see 4 pairs of bars. So the very left pair is these two algorithms in the original form, and then the next pair is using PLST with these two algorithms, CPLST and our GoAware transformation. And further to the left, we have a purple and a light blue bar, which is the performance of our baselines. Mainly two things in this figure. Our similarity representation can increase the F measure considerably compared to MSKNN in this data set, in the cross-validation we tried. But second point is that actually doing this dimensionality reduction does not really improve a lot. Actually, for our representation, it is worse, and it is slightly better for the MSKNN. However, if you look at the semantic distance, and so lower semantic distance means better performance in this case, we see that initially our method is losing to it does worse than MSKNN. And now you have the opposite effect. Actually, this reduction is helping both methods. So the bars are getting lower for the dimensionality reduction. So it helps in this case. And actually, the minimum semantic distance, at least in the way we did this experiment, was using our similarity representation with the k-nearest neighbors and our GoAware dimensionality reduction. Oops. So, in conclusion, I talked to you about two things. First, a new representation of proteins based on similarities to all proteins in our database. And that, as you saw, already gives a nice improvement in the F measure, but doesn't work that well in the semantic distance. And then a way to transform Go terms into a lower dimensional representation, and that indeed, on the other hand, is improving performance in the semantic distance mainly, and sometimes in the F measure. So we still don't have a combination that performs best or significantly better under both evaluation metrics, so we did not make the leap that we set out for yet, but still we have made some nice improvements. So I'd like to thank my supervisors and collaborators from TU Delft and from Kijin, and of course the organizers of this meeting for giving me the stage to share my work with everyone. Special thanks to the American National Science Foundation for the travel fellowship, and thanks to every one of you who are still here and listening.
Yeah, just a small side note. Your representation that's been around. It's known as the empirical kernel map, and that was proposed around 1999, and it's been used in bioinformatics for fold prediction, for detecting remote homologs, for protein interaction prediction, and in the context of function prediction. So
I know that this representation is around. I have not seen it in context of function prediction, to be honest. So I would be happy to, if you later tell me where this has been used, definitely. But of course, this representation is not new, and representing objects by distances is, of course, very widely used in many areas in machine learning. So I thought that it was used for the first time in function prediction. Maybe it wasn't so clear, but apparently that's also not the case. Okay. But yes, definitely, yes, thanks.
Okay, any other questions? All right, so thank you.
Thanks.